Welcome back, everyone. And we're close to the end of our day. Uh, so, I welcome you all to our workshop on effective storytelling. Chapin Rodriguez does not need an introduction anymore after his absolutely great talk, but I will do a very small one. He is working at the consulting firm called Creaducate in Munich. And the basic idea that they have is to make scientists reach out to other in very simple language to communicate in a more effective way. And with this workshop, he broadly wants to tell us how to tell our research story in a very visualizable way and in a story format manner. This is something I feel I keep telling to my PI, oh, we want a cool story and not just data. I don't want to just present data. We want cool story. So I'm hoping we can learn a lot out of this. So with this, I hand it over to him. I will very quickly just remind you all, you can ask as many questions as you want here. There's an ask a question feature, and we hope to keep bringing up questions every 20 minutes or so, instead of just all of them at the end. And we want it to be as interactive as possible. We will, I think, give a break in between for 10 minutes. Maybe we get to hear a piano performance then.
No.
Too much pressure.
That
We just
yes,
get a 10-minute break.
that I can't organize.
Okay, then I hand it over to him.
Thank you, Tarana, and welcome to all of you, and especially a shout out to those who are watching when it's about 10 o'clock at night or later in your home cities. I'm impressed that you're also interested, and I'm pleased, and it means that you recognize the importance of effective storytelling. I already spoke briefly with a lot of you this earlier this afternoon, and I even had interesting question from an Indian gentleman coming from social science. Let me start by saying that everything I will discuss today, I think, is extremely valid, whether it's social science or natural science, because this is Neurizons. All the examples we will look at come from the biomedical literature, but and the text that we look at. But really, the lessons are, I think, more or less universal across research communities when you're trying to reach other researchers. Because whether or not you realize it, research, and even when I was younger, it was starting this way. Research is more and more interdisciplinary, and your work brings together multiple fields. So the idea of fields, the lines are blurring and thinning, and you have to be able to express what you do in many different ways. A very good scientific communicator is able to package or present or spin, whatever word you want to use, his or her story in multiple ways. If you look at one of the key differences between younger scientists and senior scientists, is the senior scientists can, package their project for a particular conference, repackage it for a particular grant from a medical research foundation, then repackage again the project for a group of investors. And whereas younger scientists tend to be able to tell their story just in one way. It's sort of like when you were learning, maybe at high school and university, you learned things one way, your way. And then when someone asked you to teach the concept to them, you had a real problem because you could only express that concept in your way. You couldn't put yourself in their shoes and change up things so that they could see it with their eyes. And that's a lot of what we're going to talk about today. So today's workshop represents, I think, a lot of best practices that I and my colleagues in my company have collected, working with researchers from a broad range of fields: natural sciences, engineering, clinical sciences, even social sciences. There are sometimes particularities, and of course you can add in your comments if you have particular examples, if you're not convinced by something, if you have a counter example, or if you have a piece of evidence that supports what I'm talking about, a personal experience with a journal or a colleague or something. I'm going to present a lot about the structure of storytelling, my perspective on that, and we're going to look at that in writing at the beginning, and then toward the end I'll look a little bit about giving talks. And I want you to see that really the same foundations, the same approach to storytelling It works whether it's a paper, a talk, a meeting in the hallway, a poster presentation, anything. So let's get started. I mean, any of you who are doing a PhD, or even who have brothers or sisters, or who have parents, or who have romantic partners, you understand this diagram. Because that leap from between what you're saying to what the other person even hears, and getting them to process it in the way you want them to process it, that's a massive leap. And you will never bridge that gap completely. And our job today is to talk about how we can make your complex research story accessible to other researchers. You will be constantly amazed. I gave you a kind of an exercise you can do earlier today when I mentioned, if you want to really see how this works, give a talk or give a draft of a paper, show PowerPoint slides to your colleagues, to your family, whoever, and then after they've processed it for a little while, ask them to echo back the key parts of that story and see how much of the essential core of your story they can really spit back to you. And whether you're doing that with your brother, whether you're doing that with your PI or the postdoc in your lab, I think you will time and again be surprised at how little your audience really understands about what you do. And even worse, in a way, even when they think they understand, how much, in fact, they have distorted your message. And usually your audience distorts your message, favoring what they're close to, what they also research, what jargon they're comfortable with, or simply the way they see those cellular processes or regulatory pathways. And that can have more or less correlation with what you're actually doing, with what you're actually saying. So how do we bridge this gap? How do we get people to understand a bit more than what they do now about what we do? In a sense, how do we make your story stand out? How do people capture your story for what it is, not for what they think it is or for what it sounds like? And I put this example of minions, as I mentioned earlier, because at first glance, minions look essentially the same. But if you take a little bit longer and you look at them a little more closely, you see that they have differences. Well, that's exactly what that journal editor is doing when they're looking at 10 different manuscripts on their desk, all of which talk about signal transduction in cancer or pathways in neuronal plasticity. They're getting a lot of very similar sounding papers, and their job is to go in as quickly as possible and figure out the unique things that particular papers are doing. That's one thing. And then the second thing, they're going to decide if that unique thing, if they can even find it, and if they find it and it's actually correct, it's what you say the unique thing is. The second thing they have to do is decide if that uniqueness is really new for the field or useful for the field. Is it useful enough? Or is it just one more little thing no one knew, but in fact, it doesn't make a difference? So one of the first things you have to do whenever you're making any kind of research story, whether it's a poster presentation or an elevator pitch, a poster pitch, an abstract for a paper, whether you're structuring your paper, whether you're making a grant, is you need to express what I call the nucleus. And this is, for example, 2 sentences describing the why of your study, and then maybe another 2 sentences describing the what for of your study. Altogether, in my opinion, you should be able to express the nucleus in a way that's almost unique to your one study. You want to be able to do that in about 95 words. And what you may find is you'll start out doing this and it'll be 200 or worse. Then I would leave it for a while, come back, do another round of cutting and reinserting and changing. Maybe you'll get it to 100, maybe do another round. After you've done this iterative process, scientific communication takes more, to do it well, takes more effort than most of us give it. And what I want you to know is that for most people, it must be an iterative process. It's like putting on clothing. You try it on, you see if the story fits well, if it fits concisely, if it flows for different audiences. If it doesn't, you change the clothing, you try something else. And it's just like any experimental work you do, it's an iterative process. So you would hopefully iterate yourself to this lean nucleus of, say, 95 words. where you establish what are the questions you're trying to answer that haven't been answered already. And very often, all of you are working in very complex areas, very competitive areas, where a lot of people are working on essentially the same question. Well, you try to find the question or the sub-question that almost only you are doing. And then you establish why that sub-question is important for the field, how it connects to the other larger questions that are sexy and trendy. And then related to that is the what for. A strong paper or a strong grant proposal tries to define a before and after moment in the field. So what a really strong paper is the one that after you put it down makes you look at the field differently, either because you're now recognizing the real importance of a certain protein, or because you see now there's you filled in a sub-question, which now opens up an even more interesting question, so on. There should be a before and after. Something much more concrete than, our results provide new insights into cancer. That's something that can be recycled for 1,000 papers every day. You want a contribution, a what for, that almost only you can provide. And so that's what you read, and it takes a lot of time to get that. And in the best cases, you have that before you start writing the blah, right? Before you start burping everything onto the paper. And often you'll find as you start to make your PowerPoint, as you start to draft your paper, your nucleus may change. As you start to review the literature, then you realize, wait a minute, what I said here is my question. That's not entirely correct, because that actually was already answered 30 years ago. We're trying to go a little bit further. And the moments I love are when I work with researchers, and through distilling out their nucleus, they actually say, oh, wait a minute, that's actually what I'm looking at. It's not this other thing at all. In other words, they've now learned about their own project. You as young scientists realize how often, for example, I will talk to you and I will say, so what do you work on? And you will start with a very methods-based thing. You'll say, oh, I'm looking at gene X in this mouse strain. We're measuring the levels before and after this, the EMT transition, blah, 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 and it's very dense, very methods-based, a lot of jargon. And then I'm left with the question, okay, so, and why are you doing all that? And then you're forced to take a step back and you say, oh, right. We want to know whether this particular gene is involved in this early stage of the cancer transition. Okay, that I can get my head around, that I can visualize. Be very careful with that. Most young researchers tend to make the mistake of emphasizing the methods they're using, and they forget the big question. And that's where editors and reviewers and your audience want to start. They don't want to start with your methods yet. So learn to approach things from this in a problem-based way, getting to the why and the what for as soon as you can, and making it very clear as soon as possible what is the comparative advantage of your study. What are you doing that the next most similar study does? When all of you are trying to publish in high-profile journals. So when that Nature editor, when that Cell editor, whoever, PNAS, when they see your paper on their desk, their first question is not, is this good science? Because 8 of 10 of the papers that land on their desk are good science. They were done correctly. Their first question is, are they looking at something that needs to be done? And so they're going to pick up your paper and they're going to compare it not against the mass of science, most of which is not well done, or most of which is not so interesting. They're going to compare you to your competitors, to your next most similar studies in the field. And that's your benchmark. That's where you have to establish as much as possible, as clearly as possible, what you're doing that's different. Now, how do we do that? Let me give you we're going to go through some general themes, how to make your nucleus and your comparative advantage as clear as possible. One is always, as much as you can, at every step of the way, try to make your communication visualizable, imagistic, write and speak in imagery. Now, when I don't mean to speak like a poet and say, the upregulation of this gene was as vast as the plains of Tibet. Okay, Don't mean that, okay? What I mean is use language and a structure that your scientific audience can already imagine. We talked a little earlier today, someone asked, how do you make your story interesting? In a way, I have good news for you. We're here to talk about how you reach scientists better. Your audience of scientists don't need very much to be interested. They want they want a nucleus that sounds new and useful in their eyes, that aligns with what they think the major questions are, and with the methods that they think are the appropriate ones. And they want to be able to imagine your experiments. So when you're describing your experiments, when you're describing the setup that you use, the variables that you measured, instead of speaking vaguely about, outcomes or the success of the therapy, use language that they can get their head around, like survival, progression-free survival, overall survival, quality of life, self-reported satisfaction with the treatment. If it's let's see, more basic research, if you want to say that, instead of saying that this dose of the drug performed better than the other dose, or this dose proved to be optimal, help them see that. Say, this dose led to the highest viability and lowest apoptosis rate, or something like that. Your audience can imagine apoptosis, how you might measure that. They can imagine the viability and how you might measure that. And so that result, the way you describe it, it lands more strongly in the mind of the audience. Let me give you a different concrete example. Same idea, how to make your key points visualizable. Look at the upper left here. We want to identify targets in cancer. Well, you have to understand, especially for better journals, this falls completely flat. They read 10 manuscripts a day promising something like this. Now let's get a little bit more concrete. We're going to look in the ERB pathway. All right, now I'm narrowing a little bit. That's already a little bit. Now I'm imagining ERB, remembering what I learned about it, where it's involved in cancer pathways. Even more concrete, let's say we want to make a deletion strategy. Maybe we want to do gene therapy and we specifically want to delete ERB genes. Well, that you can't get more concrete. So I have the same phrase, which could be, in my nucleus, it could be in my poster pitch, it could be part of the title of my grant proposal. And just, depending on how concrete I am with these red elements, it's much more visualizable and therefore much more convincing. Your story, the more I can see it, literally see it in my mind, the more convincing it is. And it's a law of communication. So you must try to be visualizable. So all of you, especially earlier this afternoon, someone asked me about selling and using sales pitches and these very, sexy phrases like disclose new insights into the profound complexity of, or I'll tell you, here's one in cancer especially. So tumor heterogeneity. We are profiling heterogeneity, the landscape of tumor heterogeneity. It sounds okay, but editors at better journals, grant agencies, they see this day in, day out. It doesn't dazzle them. What will dazzle them are words that make them see what your angle is and what you're doing that's different from the other landscape heterogeneity paper that they just saw. That's what you want to be trying to do. We'll give you another example. This could be sentences that you'd use standing at your poster, trying to get someone to stand by your poster. It could be basically most of your abstract. Okay, so if you look at the top version, so protein trafficking is important for cell processes. Many studies have looked at trafficking using techniques such as radiolabeling and microscopy. I'm trying to get personal now. We are interested in trafficking, and we're using high speed to measure in real time. So if you look at the top version, the points of attraction here, or for an editor or the reviewer, the landing points where they begin to see what might be unique here, we have what? We have high speed and we have real time. Okay, but that's something. Now, I want you to look at the lower version. And if you look at the red bits where I've added, These are parts where I've tried to create imagery. I've tried to get the audience to imagine where we are, to picture themselves in the cell being measured by my technique. So we have trafficking. We even illustrate with a couple of examples. And these are hopefully exactly the steps that I'm going to describe in my paper or that I'll analyze in my grant. I say, okay, people have done radio labeling, microscopy, but I don't just say people have done that or they work, but not so well. No, I give you concrete if I can. Sometimes you can't, but push yourself to always do better. In this case, I can. Let's say I can say they don't give us accurate. So hopefully in this paper, I will be more accurate than those techniques. For what? I'm not going to say trafficking of various proteins or diverse proteins. I'm going to make you see the diversity. I'm going to say short versus long or unmodified versus glycosylated. Therefore, we're using high speed to measure kinetics for these different types. Now I'll say different because I know exactly what you'll visualize. You'll visualize what I just said up here in real time. Now, the bottom version is a bit longer. Probably if I had more coffee and I focused, I could make it a bit shorter even. It is true that using the techniques I'm telling you today, sometimes your text gets longer. But on average, I think you might be surprised to learn you usually get shorter. Because by really forcing yourself, interrogating every word, is this conveying concrete content, you will end up getting rid of a lot of dead weight, a lot of fat, a lot of words that you put in because you like them from other papers, but they're not actually critical. You don't need them. Here's a good example, a key protein in this pathway. So people love to say, this is a key regulator of such and such pathway. Well, how many people publish non-key regulators? Almost nobody, right? So I want you to realize that for journal editors and reviewers, when you say key regulator, they just they fly right over that. It doesn't mean anything because everything's key. Similar to the word novel. Okay, it's a novel gene. Well, maybe it was novel 3 months ago, but technically it's not novel anymore. That kind of, these are new. And when I was younger, but just so you know how young you are, when in my day, it wasn't novel, it was new. People would say new. This is a new gene, a new sequence, a new whatever, new pathway. Yeah. So everyone was saying new, and then that got overused. So then people switched to novel because it was less used. And now, guess what, folks? Novel is dying off. So I think I've already seen the next generation. It will be, for example, unprecedented. So write that down if your PIs love big words like that. So you can now say unprecedented. Or never before characterized. This is also good because it's nice and long. But you'll see I forget, the PNAS is one, and I think I forget which Nature journal. They tell you explicitly in your instructions to authors, do not claim novelty. Do not write, this is the first report of, this is the first time that someone has done. Don't say that. Why? Because actually they're responding to the same things that I'm trying to teach you about. Instead of saying, this gene is important in this pathway, make that important visualizable. Say, deletion of this gene stops the pathway. Or, so instead of saying this is an essential regulator, you could say deletion of this gene leads to 20-fold upregulation of blah, 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 or leads to dysregulation of 5 other downstream, blah, blah, blah. Then I don't have to say important, but I say it without saying the word, and I illustrate it in some way that people have no problem seeing. The more you learn to basically empower your ideas with concreteness and visualizability and less with sexy selling phrases, believe me, the stronger your writing it will be and your speaking, and on balance, the shorter it will be. So that's visualizability. That's one of our challenges. A second challenge is what I call the dimensionality problem. Now, here's the situation, and this is why communication, why I showed you that diagram at the beginning with the kind of overlapping circles. Why is communication in any context such a challenge? One big reason is dimensionality. So in research, you have your project. And if you think of your project as a three-dimensional castle, say, it's got a structure, a shape, a feel, a texture, maybe even a smell, maybe sounds. It's this three-dimensional animal. And yet, spoken and written communication are essentially one-dimensional. So when I speak, the words come out one by one. When I'm writing, when someone's reading my writing, they're reading the ideas in a linear, kind of one-dimensional flow. So what are the chances that I, as a speaker, can take that three-dimensional castle, break it down into Lego blocks in a one-dimensional chain, and have the audience receive them in a one-dimensional way and then reassemble them into the same three-dimensional castle that I had in my head? Zero. Zero. If you're lucky, the main parts of the castle will be about right, but that's it. So this dimensionality is a big challenge whenever you have a complex project to talk about. And the more complexity, the more severe the problem. Now, what does that mean? That means that you have to be acutely aware and sensitive to the very finite working capacity of your audience. I don't care if your audience are 5-year-olds, Nobel Prize-winning scientists. They all have very finite attention spans. And in my opinion, and I have a 5-year-old, so I know what I'm talking about, Nobel Prize-winning scientists can be about as attention deficit disorder as 5-year-olds. In fact, one of the things I like to say to clients, I won't convince you, but maybe I'll plant it in the back of your mind, is to think about your audience of scientists as hyper-educated 5-year-olds. They get distracted very easily. And so you actually have this problem because they can only hold, your audience can only hold a certain number of ideas in their head. They're distracted very quickly, and yet your project keeps coming at them. So now imagine, here I am, I'm listening to your talk, I'm collecting your ideas, and I'm going to reach saturation point. And if you want numbers, there's been some neuroscience to suggest that most of us can hold at any one time in our working memory between 5 to 7 chunks of information. Now, what is a chunk? I don't know what a chunk is, but it's very finite, yeah? Can you see me? My screen changed. Can you all see my slides? Tarana, you can? Okay, there we go. Great, great. Okay, perfect. Gotcha. So your audience is listening to you. Your ideas are coming in linearly, and they reach now saturation, 5 ideas, they're full. But you keep talking, so the audience now has to make room. What are they going to do? They're going to start to do a memory dump. They're going to have to decide, of these 5 ideas, which one do I drop to make room for the next one? Now, if you've set up your story right, and they know what the really key ideas are, and they know how to connect them all, then I, as the audience, I'll feel maybe comfortable dropping one thing. I won't feel disoriented. And then I'll be able to take the next idea. But if I don't know from you, if I don't know from your cues, if I don't know from visualizing your story what the ideas are or where we're going with them, then I don't know which ones to prioritize. So I'll dump something and then take your next one. But maybe I'll dump the most important idea to go forward. So that you have to be very wary of. And that's why you've got to prioritize your information. And here's the idea. Your audience is distractible. And especially your audience of scientists, because unlike general public, your scientists are highly trained analysts. So if you give them 3 pieces of information, ooh, they're excited. They're like children in a candy store. They will take those 3 pieces, run with it to build a whole story, but it won't be your story. It will be somewhere else. It will be somewhere else. So here's what I want you to do. I've been telling you, make a nucleus, crystallize out your nucleus. Another big idea, I want you to crystallize out, I would say, 4 to 5 key ideas of your work, whether it's a paper, a project proposal, a poster. What are the 4 or 5 ideas? Remember, why 4 or 5? Because that's what will fit into this working memory of your audience, whether they're Nobel Prize winners or 5-year-olds. Finite. So you're going to distill, whether your project is 6 months or 3 years, you're going to distill it down to 4 to 5 big ideas. And those ideas are going to repeat over and over in your story. And what else? You're going to take great pains to make connections among the ideas so that the audience can see what are the little phrases that connect idea 4 to 5, what connects idea 5 to 1. This is what I meant earlier when I said quiz your audience. So have them echo back, okay, what gene did I tell you I'm working on? See if they even come back with the right gene. Then say, okay, why am I working on that gene? Because most people are working on the other one. See what they come back with. Then you say, all right, I mentioned that gene, and I also mentioned this high throughput, blah, blah, blah. What's the connection between that gene and the high throughput? See what they come back with. Or things like that. See how much they get from that. In the best case, if you're very good, I would say you can hope your audience to take away maybe 80% of your nucleus, 80% of that top, your universe of 5 ideas, and maybe 80% of the connections. That's really all you can hope for. Anything else is gravy, it's extra. Which brings me to another universal rule I want to give you. Less is more. Because of the distractibility of your audience, because they're hyper analytical and are very happy to run off in their own little world with your partial information, you have to hold them in the room with you. You have to keep them synchronized on your information. So you've got to keep it simple. The more things you bring in that sound like big ideas, but aren't actually the focus of your talk, the more they will get distracted, and the less likely it is that they will hold on to really the key things for your talk. Okay? So really, this is very key. One thing you can use, by the way, I forgot to mention, the slides I will give to the organizers afterwards, and they will email it out to whoever is interested, in case you're like photographing them with your cell phone or something. You should use your cell phone for more interesting things like WhatsApp and WeChat and the whole thing. Although not right now, because I hope what I'm saying is far more interesting than WeChat. So one thing you can do, I put here a slide here. This is just a little template. Make this little matrix where you have main idea 1, 2, 3, 4, 5, and then the columns are the sections of your paper or your talk or your poster. And the task is, if it's really a key idea, a leitmotif, for those of you on the German side of the talk, it's to make sure that those key ideas are really expressed everywhere, okay, along the talk. If you have a key idea you have nothing until discussion, that's not a key idea. You shouldn't introduce anything new in the discussion. If you have a key idea that you say only in the introduction and nowhere else, what is that? That shouldn't be in the introduction. If it's not a core of your research presentation, if it's not going to be even in your results. So that's something that you can do. I saw a little request. Let me stop now for questions so far or comments.
Okay, so we can stop now for questions. I hope you can hear me. The first question is from Clyde, and unsure whether she wants to be on the screen. So I will just ask this. She asks, is it better to write a lot of papers and publish in less prestigious journals, or should one aim to write less but high impact papers and try to publish only in prestigious journals?
I don't like that question. They say that the question is absolutely, I totally understand why you asked that. When I was a graduate student, we had the same problem. I think there's no one answer. Because actually, I think it depends on what your publication list, whom it's trying to impress in the shorter term. So many, I think, will tell you that having fewer big impact papers is better because of the journal impact factor. Still, many research evaluations are done based on the impact factor of the journal. And in fact, for example, I can talk to you, I do a lot of work with China. And there, not just in China, but everywhere, but especially in China, there's a tremendous competitiveness to publish because it's the one way they can really get ahead in a very competitive structure. So they're really trying to pump out a lot of papers. And even there, now in China, as of, I think, February, there was a massive policy change where the government said, we're not going to look at numbers, at how many papers, we're going to look at impact. So they're taking various steps to force Chinese researchers to publish fewer papers, essentially, in more higher impact places. I would say it depends on who's evaluating you. So if you're applying for probably a lot of EU fellowships or postdoc positions or grants, probably, yeah, the journal impact is still very important. If you're applying for things, but listen to this, if you're applying for maybe a personal fellowship or for a grant where demonstrating your independent or your individual research capability is more important, I could imagine that it could be better to have, for example, papers in lower impact journals that are smaller and you're first author or joint first. So maybe 2 or 3 papers where you're first author and they're smaller papers might be stronger than one big paper in Nature where you're fourth author. I could imagine that could happen. And then in the application, you would explain what's going on, that you actually, you single-handedly, did all of that work. So it's a tough question. There's no one answer. You have to ask what superiors are going to look for, what evaluation committees are looking for, and they'll tell you usually if it's a grant or a fellowship, they'll tell you what they're prioritizing. But realize as a young researcher, if you're trying to establish your independence, being buried in the middle of a Nature paper, I'm not sure that always trumps having, say, a couple of first author papers in much smaller journals, but where your individual contribution is more evident. Other questions?
Yes. The next question is from Alex. He asks, how do you adapt when to point out the nucleus in your grant, application, or poster or investor pitch, as every audience or reviewer has a different interest and attitude how to address scientific questions?
Well, I wouldn't microanalyze this or micromanage it. I think I understand the question, but I don't think it's so often necessary to, you can't take account of individual biases or preferences. You can't do that. You can do your best guess. And so you think, all right, I want to submit an abstract to a conference. Who's going to the conference? Is it going to be more basic researchers? Is it going to be 50-50 basic and clinical? Is it going to be 50-50 basic and engineers? So if you know that you're going to have, for example, a significant group of engineers or medics who don't know a lot of the research side, then you make certain adjustments. But or you might ask, for example, I want to submit this to Nature, or I want to submit it to Nature Cell Biology. Well, then you might, so if you submit to Nature, Nature wants to be more generalist. So there you may want to zoom out occasionally on some key cell biology things. But if you're submitting to Nature Cell Biology, probably you don't have to quite as much. So I don't see this as tailoring to individuals and personal preference. I see it as, yeah, tailoring it to fields or groups or communities of communities of people using that methodology, communities of people asking that kind of question. That's how I see it more.
Okay, I think people really want to look at you and look at your slides, so I keep my video off.
I'm flattered. I think they just want to see the plants in my background, but that's okay.
So I will go to the next question now. And right after that, we can go back to the talk.
Sure.
So this is from Anne. She says, given that pressure to produce a story or narrative for a research project in order to get published is a major cause of irreducibility in biosciences, and that as a field of study, we need to value replication and supposedly boring research as much as unprecedented science. What's your advice on how to communicate your science effectively, but also responsibly?
Well, that's a big question. There are a lot of questions in there. So first of all, I'm very glad this came up because there's a whole movement on this. And I looked for a little while. I was briefly collaborating with an organization that's working on this, and they're still working on this. You can Google this. It's research waste as a Google term. Research waste, reproducibility problem in science, irreproducibility in science. There are a lot of researchers, a lot of them in Canada, UK, and in US that are working on exactly this problem of irreproducibility. And there was even a whole special issue of Lancet dedicated to this, and which I used to have on my shelf. If you're interested, write me and I'll try to dig it up again. And this was with a focus in clinical research, but the reality is, actually there have been, for example, consortia of researchers that have sought to reproduce landmark studies in cell biology, pharmacology, psychology, and maybe some other fields. And the results from those various attempts, initiatives, have led to the idea that sometimes 50%, so random, okay, 50% of the results cannot be reproduced reliably anymore, of these landmark studies done. And I had a friend, so I stayed in academic research, and this friend of mine, she went from academic to industry. And I asked her how things were going in industry, and she said, one of the things that she was shocked by was in industry, she said a lot of companies, if they read research papers, for example, the company wants to develop anti-cancer drug, and then they look at the literature, they follow academic labs, what they're doing, and they see that the labs are pointing to a certain molecule. They will put their industry lab to work on that molecule, but if after a brief time that their own internal lab fails to reproduce the importance of this protein, they give up, because they say so much of what comes out of academic research is not reproducible, so we don't want to waste our time and money on it. So this is a big problem. I think that there is an importance to replicating studies, and then this makes it, if you're trying to get into those journals that really prioritize innovative, unprecedented, high novelty, then you do have to carefully construct your nucleus and justify why it's so important that you do this question again. So, okay, we're going to try this one different cell line. Well, you need to justify why that, you need to repeat that study the nth time now in a different cell line. What is so important about that cell line? Why wasn't it used before? Who cares if it works in that one? What does that mean for the field? If it doesn't work in that one, what does that mean for the field? What are the stakes? You have to justify carefully. And then the last part of what I could say is, the lady mentioned, or the person mentioned responsibly at the end of the question. I mean, maybe you're getting at selling. I mean, you have to, as I was saying earlier today, I think in the long run, it's better for science, it's better for you to sell your message. If you sell something that's really well-defined and concrete, but maybe smaller, than if you try to sell hot air that sounds like it solves all the world's problems, but anyone who cares enough to dig will see that it's actually pretty small. And in fact, few people will try to even smell what is the hot air and what's the reality. They will just give up on the paper. They will stop listening, or they will reject it, or whatever. The few then who look deeply and say, but this is where your story is not this hot air, then they'll really they'll give you a hard time with their comments and so on. So definitely be responsible in what your claim is. And so what you want is a nucleus that's very authentic to what you're trying to do. And then you try to make that nucleus as convincing as you need to for that journal. And realize, and I'll end with this now, realize journals vary a lot in what is new for one journal may not be new enough for another one. And this is very often impossible to predict. What is useful for the field for one journal is not useful enough for the other journal. And it's very hard to. Your job, I would say, is to defend your nucleus as useful and unique and new as possible, that you're comfortable with, and that's it. And then you give it to the journal and you let them decide. There's actually, I think, very little influence you have on that.
Okay, so there are many more interesting questions, but I suggest we go ahead with our presentation now.
I worked hard to make this slides. If you don't let me show my slides, then what can I say? Come on. No, please. I'm very flattered, and we will see as we go. I'll try to make as much time as possible for questions. So let me give you a concrete kind of illustration of these things that we're discussing. So just as one case study, and I've given you the reference on the slide. You'll get the slides. Look up this paper. Here's just one example to look at some of these things we've discussed. So this is the primacy of beta-1 integrin activation in metastasis. And you can pick anything, but I picked this one. So let's zoom in and just look at how some of these ideas of nucleus and universe of 5 play out. So let's go to the abstract. So here's the abstract. So already from the title, you can actually see most of the universe of 5. We have beta 1 integrin, some kind of activation, and metastasis. So actually we have the top 3 things in a title. In my opinion, a good title talks about the who and the what of the study. Now, by who, I don't necessarily mean people. By who, I mean the focus of your study. In this case, I would say it's beta 1 integrin. And then the what of the study would be its activation, so the activation of that molecule. So you're not looking at its expression, you're not looking at phosphorylation, you're looking, or maybe you are looking at phosphorylation. Somehow it's activation. Yeah? And then your overall system should be in there, and in this case, it's metastasis. Now, what's not in this title is the how. So the who and the what should always be clear. The how may or may not be in your title, depending on whether it's part of your nucleus. If you're using a method that is unique, that's unusual, that you really want to highlight for the field, then maybe put it in. If you're using bog standard methods, why clutter your title with your methodology? Now let's go to the abstract. I've highlighted in yellow some things I want to focus on. So we're not going to read the whole thing. After neoplastic cells leave the tumor and circulate, they extravasate and they colonize. And that's metastasis. That's basically metastasis. Beta 1 integrins, that's our star performer, play roles in cancer, including in this thing that we're looking at, which is metastasis. So we have a link now. So we create the idea of metastasis, then we switch to a new idea, and then we come back to a pre-existing idea. So now we've embedded integrin in our pre-existing idea. So we've got a good flow here. All right. So the question is probably going to be, what are beta 1 integrins doing in this metastasis, in extravasation? And so then it turns out beta 1 integrins can be activated in this inside-out way, which it seems, however, has not been explored. So here we go. A role for this particular kind of signaling has not been explored in metastasis. So in I think it's 2 sentences. Yeah, okay, they're longish, but in 2 sentences, the authors lay out the why of their study, and they lay out the universe of 5. That's it. That's what you want to do. And the however here is a flag. As you know, that word or phrases like despite our understanding of, despite advances in, we still don't know this. That's a flag to the audience. Hello, here's my nucleus. This is my why. Here's the question I'm trying to answer. Here's the gap I'm trying to fill. And then the stage is set so that you can make sense of all this blah, blah, blah that comes in the middle. Here's where the technical stuff comes, the methods, the mess. And then we get delivered some result. And then look in yellow. So we have inside-out activation, promotes extravasation and colonization. It promotes metastasis, which suggests diagnostic and therapeutic approaches. So you can't get more concrete. We've closed the circle. The what for of the study, the second part of the nucleus, loops back to the first part of the nucleus, and we close the circle. And we have a concrete image of maybe we can target this activation of integrins to stop metastasis. Maybe we can look at activation status as a way to diagnose if the cancer is going to metastasize. You can't get more concrete than that. Much more powerful than our results provide new insights into the role of integrin in metastasis. That will fall completely flat. Many integrin papers could use that. So we've seen the title, we've seen the abstract. So look, here's our world. If we were to do a plot of the big ideas, it might look something like this. In fact, I found just 4 here. Colonization, extravasation, integrin, and inside out. And what we want is, as quickly as possible, the reader should leave the room after reading our paper, after hearing our talk. They should leave the room with those 4 ideas in their head and these short explanatory phrases that put those 4 ideas together into this diamond, pentagram, whatever thing you want to call it. Okay, that's what we want to do. That's our goal. And if you can achieve that, you're doing very well. Now, one trick to help, that's in the title and the abstract. So we need to maintain that coherence, keep our universe focused on a finite number of key ideas in the introduction. And then we're going to go into methods, then we're going to go into results. How do we do that? One trick, which is very common in life sciences, so some of you may already do it, I call it thinking out loud. This is from the same paper, this integrin paper in metastasis. I've highlighted in yellow phrases where you're literally, they're like signpost sections. You're telling the reader or the listener, this is where we're going. So before I barrage you with lots of method details, with concentrations, molarities, drugs, group A, group B, group C, let me remind you what this is all for. And so you have, statements of purpose to begin to address. You might have introduction, actually. You might bring in old knowledge to set the stage for your next experiment. So integrin requires interaction, da-da-da. Therefore, we, da-da-da. And then the following sentences lay out that experiment. Here's another example of that. By the way, all of these text blocks come from the results part of the paper. And so you might say, but look at this. They're reporting introduction in results. Absolutely. I want you, when you're storytelling, whether it's a paper, whether it's a talk, I want you to get away from that traditional idea that, okay, introduction is past knowledge, methods is what I did now, results is new knowledge, and discussion is, future. Forget it, doesn't work. I want you to adopt a more dimensionality-based model where you say, okay, I have these Lego blocks now, and they're filling up in my reader's mind, and my reader is dumping blocks to make room for more. So that means that if I said something in introduction 10 blocks ago, and I need them to think about that right now for this experiment, I might need to repeat that block. I'm going to reinsert it into the pathway. Realize that good storytelling requires repetition, but strategic repetition. So you're not going to repeat yourself so much that your audience says, my God, this guy's a robot. You're going to realize, I need spatiotemporal correlation. If I want my audience to associate two ideas, I need to put them together in space and time as much as possible. So if I've got the key idea here and a key idea 3 minutes ago, and in between I have several other key ideas, forget it. Because the audience is going to take the latest idea, try to match it up with the last one they heard, but that's not it. They have to go back many key ideas to get to what you want. So be very aware of that dimensionality problem and how distractible they are. You have to be very aware of that. So part of that repetition comes even in the macro structure of a paper or even a talk. So in my opinion, a well-written paper has what I call signpost sections. These are regular points of the journey where you stop and they're breathing space for the audience. This is where you say, okay, let's take stock of where we are. This is what we're trying to do. This is what we know. Let's go. What do we need in order to go forward? Blah, blah, blah. And so these are the main parts of a paper where you should really consciously write your nucleus, essentially. These are places where so I want you to go to papers in your field that you consider well written. And I'll bet you money that if you look at those parts of the paper, these signpost sections, you're going to find that nucleus repeated over and over again. Probably in a way that you don't quite notice because they change a few words. And that's all of you need. Now, here, that's one of the most powerful things you can do to be interesting and to hold your audience. Okay, we'll come back to this, I hope, later when we talk about oral presentation. So same when you do a talk, periodically stop, synthesize, tell your audience, this is where we've come from. This is where we're going in order to get there. This is what we need to do. Here we go. And if you can do that, you'll be well on your way, I think, to making things more understandable. I gave you a challenge earlier. I said, when you draft a paper, when you make a talk, prepare it in front of a colleague, and then force them to echo back the nucleus, force them to echo back the universe of 5, echo back the connections among the 5 things. Same assignment here, I call it the elevator pitch. Some of you may have heard of this. The elevator pitch is an idea that started in business. And to be honest, I think a lot of the rules of good communication come from public relations and business communication. And the problem is, so I have tried to go into the literature and look at what neuroscience is telling us about the way the brain processes information, because what I'm giving you in this workshop reflects how, the way I see it at least, just reflects how people process information, whether those people are neurobiologists or whether they're microbiologists or someone from the street. Okay, doesn't matter. Most Homo sapiens process in these common ways, and that's what this workshop is crystallizing. And the problem is, because science has taken so long to accept, I guess, how people really process information, we haven't adopted what the business community and advertisers have been doing for decades. So if you know, unfortunately, the literature is basically mostly about advertising. Hopefully this will change, but adopt some of their, you know, advertisers know you have to get an audience that has a short attention span, very distractible. You have to get them interested. Well, that's what we're talking about too, because that's your audience as well. So try to make an elevator pitch. This would be 1 minute, maximum 90 seconds. Try to shoot for about a minute, where you express your nucleus as well as the methods and results details. So maybe put first part of your nucleus, the why of your study with the gap. Then you give the methods you used, give details, whatever few key details are critical to get the big story. Then give your results, the few key results, not all of them, that are key to get the whole story. And then close with the what for. How does your work define a before and an after in the field? Try to do that in 1 minute, 1 minute, 10 seconds. I guarantee you can do it all very well, but it will take iterations. The first time you do it, it'll take 3 minutes. Then it'll take 2, then it might take 1. I've done this over and over in workshops with bioscience, biomedicals, clinical researchers. And what's really impressive is when they do this exercise, they distill it down, they're happy with it, and it makes them realize, that's actually what the purpose of my project is. It's not that at all. It's not what I came in the room thinking it was. And that's a nice moment. So try to do this, practice it with and that elevator pitch of 1 minute is then brilliant as the opening for your talk, even as the close for your talk, as your poster pitch when you're standing there and you have really short time to convince that person to keep standing there, to go through all of your beautiful immunocytochemistry and whatever. That elevator pitch can work very nicely as that core. So definitely I encourage you to practice that. And then with that, I come to the end of this part of the talk of fundamentals, basically macrostructure. And we're going to take a very short break. And then in the second part of the talk, I want to look actually at some specific things about language, microstructure, where you put words, how you put words, that especially for English can be extremely powerful, but I think for any language in which you're communicating science. So I will stop and see if there's any questions or comments.
Okay. So I hope people can hear me. One question we have from Alex is, where do you draw the line between stating limitations and bashing scientific contributions, as you need to mention references as examples of the limitations? Tricky.
Yeah, it's a great question. In a way, it's an easy question to answer. I know I've been through this. I'll give you a quick story. I remember, so I published a paper on an enzyme, and we did the crystal structure, and then we proposed a mechanism. And I was proud. I love going to libraries. Okay, this was the old days. Okay, when everything was not online, so I would go into the library and read papers. And we published this paper, and we proposed a mechanism. And some month later, I get an email from an extremely angry professor in Russia. And the professor said, "You didn't cite my paper. I also proposed a mechanism." And me, I'm the young graduate student. I'm terrified of pissing off my competitors or colleagues or whoever. Oh my God, I offended someone. What have I done? Chapin, what's wrong with you? You looked at the literature. You know that literature, right? I have to write a thesis. I have to write everything in the literature. And then I realized what that paper was. And that paper was like half of a page. It was in some really bad journal. The figure showing the mechanism was hand drawn Really, I can do better, and I'm no artist. And then I said, okay, I don't feel so bad that I didn't cut, because I didn't even think that this was even a mechanism. They just drew a thing with something sitting in it, and that was it. But in the next follow-up paper that we published, I did mention that paper. So yeah, you do want to be careful. But in a way, your question is easy to answer, because you don't make it sound like other studies are limited because they're badly done. You state objective gaps. So, for example, these experiments have yet to be done in this cell line, but no one can argue that, right? Or you say, or you spin it with a more positive tone. You say that previous work has shown us A and B, but it's not clear whether this is also true in C. And that's also, by the way, a good way. If previous papers reported C, but you and or other people don't really believe it, you can say, well, there's something about C, but it's still kind of unclear. It's controversial, right? So you can yes, you need to be diplomatic, but basically keep it objective. Just point out gaps that are absolutely clear, okay? Now, let me give you let's give you an example. Let's say other studies have looked at 200 genes. And now you're coming along with essentially the same study, and you look at 1,000 genes. Now, instead of saying, okay, previous work looked at 20 loci involving 200 genes, or you could say previous work looked at a limited set of genes, and then you think, oh no, wait a minute, 200 is still a lot, right? Depending on the question, on the field, whatever. Why don't you just say, okay, look, that article looked at 200 genes, but that didn't include these two sets of genes that are involved in these signaling pathways, which have been linked to the disease. So in order to do a more systematic screen, or in order to be sure to include specifically those pathways, we looked at 1,000 genes. Do you see the difference? So just make it more objective, and that's going to force you to be very concrete about your specific comparative advantage. What is that little step that you're taking that you want to say actually makes an important, useful difference to the field?
Beautifully answered. I
hope it's helpful.
suggest people are already thinking of modifying their poster speeches, so I believe so. I suggest we can take a break now
Okay, great.
for 10 minutes.
Perfect. Yeah, so maybe at
And
6:15 I'll come back.
Yeah, 6:15
My
German
time.
time.
Yeah,
Yes.
yeah. And the rest of
Okay.
you, I'm sorry. So go drink some
10
coffee really
minutes from
fast.
now.
Okay,
Perfect.
sure. Bye-bye.
Bye. Okay, are
Okay,
we ready to start?
there we go. Are we?
Okay,
Yes. Okay.
perfect. I will disappear again so that people.
Okay, thanks.
Okay, perfect.
All right, so let's go. Here we go. All right, so let's go to the next part where we just look. I'm going to take a few slides. This 2-hour workshop is a kind of a crystallization, if I can use my own word, of a lot of the things we do with our clients. And so I don't have time to present everything, of course. So I just want to present a few slides that are very specific for language. So we've talked about structuring the article, some aspects of structuring an article or a talk. Let's now zoom into individual sentences a little bit and look at the language that you can use. Most research communication is happening in English. So in particular for English, maybe some of you who have some other language as your mother tongue, sometimes you feel tired of English because everyone's English is so great, English is everywhere, and then you think, well, okay, English is not the savior of the world, and it is true. I think compared to some other languages, English can be pretty inflexible. The thing to realize is that more and more of the audience that you have, and more scientists are using English to communicate the research, and a smaller and smaller percentage are actually native speakers. So in particular, because of that heterogeneity, I want to encourage all of you to try to follow this golden rule to speak or write as subject, verb, object, where you put the subject and the verb as close together as possible. And this suggestion, as well as following suggestions, I'm showing you with examples from English, but I do believe that if you incorporate a lot of these suggestions, on average, your writing in any language can become more concise, less kind of fat heavy. So I'll show you examples later. What we do is several things with this. By putting the subject and the verb as close together as possible, we help the audience associate them together. Remember that spatiotemporal correlation has to be there if you want the audience to associate your ideas. The other thing we gain is we present the frame of the sentence at the beginning. We prepare the reader. And then that object that comes at the end, which is usually what's full of molarities and drugs and figure numbers and p-values and so on, that comes at the end after the frame has been set. So before the reader or the listener encounters those very dense information, they already know what the general story is about. This sentence is telling me about the results, or this sentence is telling me that group A did better than group B. And then even if I don't understand the exact statistics that come or some of the jargon later, I understand that group A did better. And so at least I have that idea. So let's look at some examples. The other thing, related to this. Sorry, you shouldn't take a break. I lose my focus. So the other thing that we accomplish by doing subject, verb, object is we reduce the risk that the reader has to go backwards. So as a universal rule, whether you're speaking or writing, try to tell your story, try to communicate so that your story is unidirectional, that you never force the audience to loop back and say, wait, what did he just say? Or, say things like, on that thing that I showed you on my previous slide, no one's going to remember what you showed on your previous slide. You'll be amazed how quickly it's in and out. Remember, the Legos keep coming, and we keep doing memory dumps, so people don't remember what you said 30 seconds ago. So we're going to prevent the reader from going backwards. Look at this example here. Let me take my breath because it's a long sentence. The level of apoptosis with PMA was significantly higher than with CZ1 based on Duncan's test after correcting for baseline differences due to cell cycle asynchrony 30 versus 15%. Now, if I ask you quickly, that 30% refers to what? The 15% refers to what? Right? Time's up. Too many seconds, okay? Most of you probably have to read that a second time, if not a third time, to see what's going on. There's no reason for that. It's not such a complicated idea here. So what can we do? Create that spatiotemporal correlation. So what I'm going to do is rewrite this. And actually, my slides are a little off here. The red line should be under respectively. I'm telling you not to make your audience go backwards. be very careful, almost always avoid respectively, because respectively is particularly horrible. It makes you go backwards and forces you to compare actually 4 things, 2 pairs. So it's really death. So there's almost always, maybe 1%, there's not a way that you can rewrite to avoid making the person go backwards. In this case, what I'll do is I'll get rid of respectively, and I'll just put the numbers immediately after the drug that they apply to. So little things like this, where you're consciously thinking how that audience is linearly processing, Legos, blocks coming in, can really add up over the sentences of your text or in your talk. Let's look at these 2 sentences. Imagine that these are sentences one after the other. So first sentence, the test and control groups were compared. Next sentence, haplotype A frequency and HEW coefficient were higher and lower in the test group, respectively. Now, as you can already imagine, okay, what was higher than what? What was lower than what? Time's up, right? Here, so respectively is our problem. Now, I told you earlier, I want you to think of your audience as hyper-educated 5-year-olds. Let's treat this like a 5-year-old would. So what we have is we have one outcome, which I'll color in one color, and that's higher. But actually, there's another outcome in green, and that's lower. Now, if we go linearly, we have yellow, then we switch to green, then we switch back to yellow, then we switch back to green. As a 5-year-old can tell you, that's just a mess. So be conscientious of your audience and say, let me put the yellow with the yellow and the green with the green. Think like a 5-year-old. So how about something like this? So now we have 2 colors with each respective partner. And I'm still not satisfied because I'm a nerd and I have to have it perfect. So can I make things even tighter? I can by doing subject, verb, object. I'll start with the subject in this case, the test group. And that allows me to put the yellow immediately next to the yellow. Now this is a minor example. Imagine if this phrase were actually longer and complicated and had some big ideas in it. Then it's really important to be able to do something like this, where the higher goes immediately next to this word. And I accomplish that by creating a subject here, showed da-da-da-da. And I don't have to mention the control group because the previous sentence already said that my comparison is between the test and the control group. But I can guarantee you, if you learn to write or try to write consciously, more like this, compared to this, the effect is exponential. I'll say in a non-mathematical way. It's cumulative. The effect is highly cumulative. The more you write like this, the shorter your sentences will be, the faster the text will flow, the faster your story will flow. It will seem less heavy and less bogged down in its own detail. And then the audience will wake up. They will get less kind of, oh, this train is moving so slow. Let's look at another example. The upregulation of CARC10 as a result of MAP kinase signaling in the presence of elevated levels of PMA can cause hyperplasia. Now, do you like that sentence? I don't have to see your faces to know you probably don't like that. Why? The organizers even don't like it. That's a good sign. Why? Because it's sending me backwards. Look at what they're doing here. They're relating a causal chain, cause, effect, cause, effect, cause, effect. But they're not doing it in a way that responds to a causal logic. So when you, and especially to those of you, which is probably most of you, who work on signaling pathways or cascades or things where A goes to B, goes to C, goes to D, goes to E, right? Be very conscious, how can I best tell my story? How can I take advantage of maybe the structure of my system, of the temporality of my system? First A happens, then B happens, then C. What natural logic or organizing principle can I use to narrate my story? And follow that principle. Don't just write it the way you've always written it or the way that you paste it from another paper. So our problem is here that upregulation is actually the effect of MAP kinase signaling which itself is the effect of this, and then the ultimate effect of everything is hyperplasia. But let's narrate it more logically. I want to do it like this. Don't send anybody backwards. How about high levels of PMA stimulate MAP kinase, upregulating CARC, leading ultimately to, and you can put a phrase in here because you're conscious that this chain is now getting a bit long. You could say ultimately leading finally to, leading in turn to, something to say, I'm sorry guys, but I had to write this all. You could even break this up. You could put a sentence here, period here. You could say high level stimulate, upregulating CARC10, period. This leads to hyperplasia, or the result is hyperplasia, something. You could break it up. Often people ask me, what should I do in this sentence? This is hard to understand. Very often the quickest cure, it may not be a complete cure, but a quick therapy is to put a period, to break it up. Why is that? Because your audience saturates so quickly. That finite working memory. Keep it simple. 1, maybe 2 major ideas per sentence. 1, maybe 2 topics per paragraph. When you change, put a new sentence, put a new paragraph. Here's one test I would suggest that you should do. When you draft something, read it out loud. Read it out loud, I'm not kidding. And if you are running out of oxygen and you can never finish your sentences before you run out of air, maybe they're a bit too long. Okay? So if you, in my opinion, the more that you write papers, the way that you might speak, it's better. Now, when I say the way that you might speak, I'm assuming you don't speak like, yo, beautiful day, let's go out and eat, hey, let's play. Well, I'm not, hip, super hipster, whatever, however you young people talk nowadays. I don't mean jargon and slang and informal. Talk formally, talk scholastically, academically. But give breathing points, rest points in your talk. So, and the same when you speak. Probably some of you have been in talks, presentations, where the speaker, even though they're speaking, they sound as if they're telling you what they memorized from their paper, that they're almost reading their paper word for word. That will put people to sleep. That will lose your audience. Not anything else about, do you have animation on your slides or colors or whatever. All right? So be very mindful of that. Give your audience breaks. Give them that, and then periodically summarize. Give them that opportune repetition. Here's 2 texts. I'll stop talking. I want you to read both of the texts and tell me if you prefer the upper text or the lower text. Do you prefer upper text or lower text?
I've asked them to write in the chat.
I saw that. They don't know. They haven't decided. I'll give you a hint, people. It's the same text. There's just a slight difference. All right. So look. Lower. Oh, thank heavens. Okay. There's usually one in the group who writes upper. Every time I do this in workshops. Lower. Okay. And now the ones who were going to write upper, they're not going to type. I see. Oh, okay. Very good. That was our informal poll. Lower. Oh, you're too easy. You're an easy crowd. Everybody said lower. No one said upper. All right. What's the difference here? It's that spatiotemporal correlation. So the second sentence starts with the idea of discrepancy. Okay. The previous sentence ends with it here. They're very close together. That's called a relay. The first sentence ends with the idea. The second sentence picks up that idea and moves further. This is a very nice way to make a transition between 2 sentences, as well as between paragraphs. So you end one paragraph with the idea, and then you pick up an aspect of that. You just continue in the next paragraph. All right. Here, in contrast, in the upper one, we have the idea, and then we introduce various other things that might seem important. They're big words, jargon, right? Then we come back to this discrepancy. Now, what is your audience going to do? Most readers will immediately start to map backwards this discrepancy. Does that refer to this? Does that refer to probe length? Does it refer to washing? Does it, right? And, you know, valuable seconds even are lost here. All right. Someone wrote, I didn't notice any difference. I like them both. Well, I gave you a relatively easy example. All right. But what I do want to argue is, so in this case, they're not so different, right? But there can be cases where this is critical. And whatever is true, the more you write more like this, making relays between sentences and paragraphs, the tighter your writing will get, and this will have a strongly cumulative effect on what you're doing. All right, let's look at these sentences, which are real, by the way. I modified them slightly to prevent people from getting in trouble. The model with the smallest residual following eigenvalue optimization and a maximum likelihood P was accepted. Now, what's the subject here? Model. What's the verb? Was accepted. Look how far apart they are. Now, this sentence, you know, the English is fine. And the second example down here, the English is fine. We're not here talking about grammar, by the way. These slides I'm showing you, if you'll notice, they're not about English grammar. They're about structuring your words, what you say when. And as I mentioned, I believe that following a lot of these tips can help you whatever language you're talking in. Now, this sentence up here is perfectly fine. And in this case is not so extreme. Just realize how much more processable, how much more quickly processable it would to say something like, we accepted the model with the smallest. And what I'm doing is I'm doing subject, verb, and then a long object with residuals and eigenvalues and p-values. What am I doing? I'm getting the frame out of the way. I'm saying whatever that's coming, however complicated it is, we accepted that. So you might want to pay attention. If you wrote, we rejected, maybe the reader can decide if they don't understand the object at the end. They don't have to, because the authors didn't use that anyway. Here's a more extreme example. Same illustrating the same thing. I only changed a few words in this. It's real. Though detailed considerations are beyond the scope of the paper, studies in which the stress-induced transcriptome rather than baseline profiles are the focus, and in which HEC-dependent epigenetic... Okay, I don't need it. My God. What is the subject? Studies. What's the verb? Should be undertaken. Okay. Now look at the distance between them and look at how many big ideas fall in the middle and can throw off an audience that's trying to understand. I'm sure that anyone would have to read this twice, maybe 3 times. And then imagine that you read that and you spend at least 30 seconds reading this, and you come to should not be undertaken. Imagine how pissed off you're going to be that you just wasted your life reading about studies that aren't even recommended. So how can we avoid that? Let's put subject, verb, object. Let's do the frame at the beginning. So for example, we recommend studies in which, or we recommend designing studies in which, and then comes all this blah, 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 blah here. So I get the frame out of the way. Whatever's coming, we recommend it. So you can decide whether to work your way through it or whether not to. You want to be just conscious of your audience, their limited processing time and attention. Last comment on that point. These lessons we're talking about go even to references. So some scientific journals will use not numbers, like 1, 2, 3, 4 for references. They will use, I forget what it's called, the Vancouver, Harvard style or whatever. They'll use the author's name and the year. But this can also create problems. So look at this text. Although other outcomes such as thermal inversion, hysteresis, or retroconsumption are not infrequent. I guarantee you by the time a lot of readers get to this, they're going to say, wait, what was not infrequent? So even though you think they're references, they're not doing anything in my story. They are doing something in your story because those are Lego blocks that are getting in the way of the other Lego blocks. So what can I do? What can I do? One thing I could do is I could put all the references at the end of the sentence. And I made that comment in a workshop, and the student where this work came from, he said, no, I can't do that because the people in my field, right, I have to put a certain name with a certain, a certain name with a certain outcome. They'll notice if I just group everyone together, they'll say, no, I discovered that other outcome, not this one, blah, blah, blah. Okay. then maybe you still have to keep it split. But what I'm going to do is I'm going to finish the idea before I get into the blah blah. I'm going to say, although other outcomes are not infrequent, such as this one, blah, this one, blah, okay? So I finish the frame before I get into the blah. So just at this level of thinking about being conscientious of the audience and what they can process, this can really make a difference. And with that, that sort of closes our discussion. We've looked at macrostructure of papers and talks. We've looked at even microstructure, a little bit of how you structure the words in your English. The last part of my talk, I just want to talk about talks, actually, if I can use that word. I see someone wrote a question. Would it be better to say other outcomes are frequent? Yeah, sure. Look, the not infrequent is a mainly British invention where you don't want to say that the outcomes are frequent, but you don't want to say that they're rare. So it's a kind of a subtle trick. It's a subtle trick. It's a subtle trick. All right, so let's talk about talks. We could do a quick poll and see how many of you like to give talks. Usually, I don't know, 30% do. Oh, someone's laughing at the Brits. Don't laugh at the Brits. They mean well. Where was I? Scientific talk. I did my PhD in Britain, so I'm partial. So talks, most, I would say maybe 35% of people like them, and the rest don't really like them. You get nervous. Remember what I said earlier, I think? If I said it with this group, I can't remember. Nerves when you give a talk. Oh, yes, not unwell. Perfect, indeed. Nerves when you give a talk, realize that I think they're largely a form of egotism, self-centered. You get up there, you immediately assume the audience is judging you harshly, of course. They're never judging you positively. So there have been, you think that they're looking at your clothing, your shoes, your accent, your slides, everything, and you just start to shut down. You just you deer in the headlights and you freeze. The reality is the audience doesn't care about you a whit. There's a Britishism for you. They don't care about you at all, and they just want your story. And that's why actually I encourage you to practice that nucleus and use that as the first sentences of your story. And then boom, you have them very soon, maybe in a universe of 5 already on the table. Let me give you some general thoughts on talks. The structure of your slide, the content of your slides, the way that you tell your story, I already talked about. You're going to make a clear nucleus at the beginning. As you go through the talk, you're going to periodically stop and give the audience signpost sections. Now, why is that important? All right, you're scientists, so we're going to make a graph. We're going to plot the attention of your audience as a function of time in your talk, okay? Now, at the beginning of your talk, the audience is very alert. As you know, think of when you go to conferences, the next speaker stands up and the moderator says, okay, and now Jonathan Williams from Imperial College will talk to us about whatever. And, you were listening to me earlier and thinking, wow, this guy says that I should treat my science audience like hyper-educated 5-year-olds, but my audiences are way smarter than that. Well, you people, consider the following. If scientists are so smart, why is it that after they've suffered through 20 PowerPoints and then number 21 gets up, everybody still perks up and says, oh, no, this could be exciting. This one will be different. I'm sure of it. And then after 2 sentences into the talk, they realize, oh my God, it's the same. And then number 22 comes, oh, this could be different. So actually that works for you, because no matter how bad everybody else was, when you get up and you come to the microphone and you open your mouth, people are strangely really interested in what you want to say. So the beginning of the talk is easy. That's where you have their attention at the most. And that's why a lot of people are usually most nervous at the beginning, because you know that they're interested, then they're looking at you. And you can't wait to lose their attention and feel safe again, but we'll get to that. So because their attention is maximum at the beginning, because your nerves are at the maximum at the beginning, do consider, really, don't memorize your whole talk at all, but do maybe memorize the first minute, which, by the way, can be that nucleus, that universe of 5, that comparative advantage. Get that set, make sure that's smooth, that it's really refined and concise. Now, that's the beginning of the talk. At the end of the talk, again, the audience is with you, and they're so happy. You know how it is. You suffer through a talk. You're like this. You're staring at your phone. You're wondering when the coffee break is. Can you get out of the room, please? How much longer? And then the speaker says, and now for my last slide. Or they say, to conclude, or maybe they're really take pity on you and they say, okay, just 3 more slides. What do? People perk up. The antennae go up. Yes, yes, I can pay attention. So everyone is staring at the end because they want the take-home messages, no matter how much they missed, which is most of it. In the middle, they want that take-home message. So you don't have to do anything for the audience's attention at the beginning and the end. So certainly use it. The problem, folks, if you can guess, is in the middle. I call this the black hole of the middle, okay? So someone wrote, this graph doesn't hold true for this particular talk. That's very sweet of you, and you're lucky I had espresso before we talked. So, but imagine the irony of this. So look at, it's the middle of the talk where usually you lose everybody. And yet, what do you do in the middle? That's your meat. That's your methods, your results. That's everything that makes you unique. So this is horrible. How do you keep people with you in that black hole? One major thing you must do is break up your story into digestible pieces. What we've been talking about all this time. Figure out, universe of 5 major ideas. Repeat those constantly throughout. Interconnect them. Come back to them. Make them recurring. Use opportune repetition. Stop periodically. Summarize. Divide up your experiments into blocks that look at A. Okay, then summarize. Now we've seen A, but A raises the question of B. So make a tight link into B. Then here's B. Do B. Then summarize at the end of B. And then say, how do you go into C? And so on. Break it up. Try to make a thread, okay? Try to set up each slide in a way that the audience can almost predict what is coming next. Those are the tightest stories. You already know what's coming next because of the way that the speaker has set it up. And less is more, right? Whether it's a talk or a paper. So just be very wary of that. So speaking of slides, I'm not going to say very much about slide design, except what I think is actually, the most important, and that is simplicity and clarity. Less is more. Keep it simple. Be careful with colors. If you use 5 colors, you can count on the fact that the projector, no matter how new it is going to screw up 1 of them. If you use 7 colors, it's going to screw up 2 of them. Clarity. We said that you should communicate so that your audience never has to go backwards. Well, if they're reading your paper, they can go backwards if they really need to. But in a talk, they cannot. So once I drop that Lego block, it's gone. And believe me, I will not struggle to pick it up again. So be very wary. Repeat yourself if you need to. Make every slide a complete story. So start with, this is what we're trying to do in this slide. This is how we did it. This is what we saw. This is what it means. Now it raises this next question. And then you go into the next slide. So now we're going to ask this question. Here we did it. This is what it means, and so on. And then maybe after a few of those slides, you synthesize everything, and then you somehow go forward. And you do that constantly. When you go to conferences or stream conferences, look at the difference between plenary speakers and the younger scientists who are getting their first 10 minutes of fame at a conference. Very often the difference is that those young ones are so terrified of establishing themselves in the field, of establishing how hard they worked and how smart they are, that they speak fast, they pack the slides with every experiment they ever did. Whereas the plenary speakers who are presenting 20 years of work, you see that they use 2 Western blots to summarize 5 years. And they don't have any problem with that because they're really into the story. They're going to tell you a story. I want to encourage all of you. I know that you're working against that pressure that you have to prove yourselves, but just realize that less is more. So if you yourself know that slide, when you put it up, is going to make the audience say, ah, right? If you know that yourself, then do the same thing we do nowadays with papers. Say, all right, of those 12 Western blots, maybe 8 of them can go in supplementary, and I'll just focus on these key ones. And so this slide where I was going to talk about 12 Westerns in 2 minutes, I'm just going to talk about 8 Westerns in 2 minutes. The other ones I'm going to put after my thank you, so that if there's time during the question and answer, I can come back to them. So don't hesitate to do that. I used to do that all the time. I would make slides after the acknowledgments, the thank you slide, and they were always ready. And guess what? I never used them. That's the sad truth. You never use them. And in the process of playing with yourself that, oh, I'll still show all those Westerns I ever did, you end up making a concise main talk. One thing about slides, keeping each story, making each slide a complete story, please don't use abbreviations or use them minimally. Okay, in a paper, you think, okay, I define the abbreviation here, and now through the whole paper I can do the abbreviation. Well, in a talk, that doesn't work because no one goes backwards in your slides. So try to find short, informative names, spell things out. There's, in my opinion, almost never a need to abbreviate unless it's the protein that you're mentioning, 50 times a minute. Okay, but for groups, if one is control or whatever, maybe put informative names that don't require me to read a legend, and certainly not a legend that you mentioned 3 slides ago. All right, let's look at this slide. Some of you make slides like this. You don't realize it, but you do. Typical, too much text, right? Too heavy. Your audience are hyper analysts. If you give them this much text, what do? Your audience starts to read it from left to right. You go into robot mode. You, in fact, might start to read this left to right when you're presenting it to the audience. But you compensate. You know that pictures of babies and cute animals get a lot of attention. So you balance the heaviness of the slide with a cute animal. It doesn't work, okay? Especially in the long run. I know what you're thinking. You think, I'm ahead of this guy. This is so 90s. I do it totally differently. This is what I do. So you're going to tell me that you put bullet points. Look, folks, it's pretty much the same effect. Yes, there's some white space, so there's a bit of breathing, and there's still the dog, but it's still long sentences. Most of the time, they're even complete sentences. And most academics, most of your audience, if you give them a complete sentence, they will start to parse it from left to right completely. All right? And that's the problem. Because when I'm parsing that slide, when I'm reading it like a piece of text, I'm not listening. And I'll show you evidence in a second why that's death for your presentation. So what do I want you to do? I encourage you, less is more, as much as you can. For example, use very short fragments, very short phrases. If I show you this slide, probably most of you know immediately that if you're going to make sense of this, you've got to listen to me, at least some of the time. So that's what's going to happen. But if I show you a slide like this is the famous conclusion slide. Most people's conclusion slide looks exactly like this. I hate to tell it to you people, but when you put that up as your conclusion, no one is listening to you. They're so excited to finally get your take-home message that they shut you off completely and just read for themselves. Why do that? If you're going to do that, show this slide and then shut up. If you really want them to listen to you, show them something that forces them to listen to you. What's going on here? Let me shoot ahead. I'm going to skip to, What we're talking about here is competition in attention. We're talking about the fact that your audience is divided constantly between you and your slides, and they are switching like mad. And most speakers, they actually compete with their slides. They don't complement their slides. So the audience is left to decide to whom they should listen, because the audience cannot listen to both channels well. So if the speaker is talking low, is talking boring, I'll pay attention more to the slides, which means I'll miss whatever they're saying. All right. If I'm talking more animated, more interesting, you guys were writing in your comments earlier, you said, show us the image of the speaker. No, show us the slide, show us the speaker. There's a competition going back and forth. That's happening all the time in your audience. And so what happens? I say a key piece of information, but you don't hear it because you're on the slide. Or vice versa. I say something else and you miss the key information on the slide. If you don't believe me, here's a piece of evidence. You can look in the whole literature, but I'll save you the time. So this is just one study where they put healthy people, men and women, into a booth with a chair looking at a computer screen. And the people had to look at the letters and numbers on the computer screen or listen to them in their ears. And the researchers told them, okay, first we want you to focus on the screen. You'll be quizzed. You'll get a memory quiz on the numbers and letters that you see on the screen, not in the voices. In a second trial, they were told, okay, we want you to focus now on the voices you hear. That's what's going to be on the memory test. And of course, what did they see? The more attention people paid to the screen, the less attention to the auditory processing, vice versa. And the conclusions of the authors is attention is limited. It's a zero-sum game. No audience can simultaneously attend well to both channels. So what I want you to do always is complement your slides, not compete with them. What does that mean? Again, so if you're going to show this, right? If I want you, to be here, then I'll use my laser or my arrow to circle it. I will say exactly this word at the same time as I'm circling it. If I want you to pay attention to me, I will turn my body and face the audience. I will not give you any clues pointing to the screen. I will say my message. Then I want you to go back to the screen. Okay, I'll turn my body and put my laser on whatever it is, and so on. You need to constantly redirect and synchronize the attention of your audience where it needs to go. Because if you don't do that, and 95% of speakers don't do that, the audience will do it for themselves. And they will decide when to do it. And that will be the Gaussian problem. So at any given moment, 10% of the people are reading this, 15% are reading this, 70% are on the dog, and however much is left from 100% are looking at their watch to know when this is over. You want to synchronize your audience on exactly that piece of information where you are with your voice at that time. That's the best way to do this. And to be honest, I'm anti-slide. I'm really anti-slide. And let me give you guys have been asking me, or I've been giving you advice on tests you can do for your own communication. Here's a test. Next time you have to give a slide talk in your lab group, for example. I mean, you can do this with a 5-minute talk. Give the talk with slides, normal, like you always do. But then give it without a talk. And have some audience of friends, have some beer nearby and popcorn to make it a little bit less painful for them. Have them tell you which version is better. And I think you might be surprised to know how often the one without the slides is better. I remember a couple of workshops where I did this. So the researchers, they're all PhD students, like a lot of you. They had to give a 5-minute research talk with slides. So, of course, they're, 5 minutes. That's so short. What can I say? And then they would come with 15 slides and pack it in. But then I forced them, okay, now you have to give the talk, and at least of those 5 minutes, at least 2 or 2.5 had to be without slides. So what did people do? Maybe when it came time to talk about the one key result and the implications, they turned off the slides and stepped in front of the screen. Or when they had to present a mechanism, or they had 2 competing mechanisms, and they had to present them and explain the discrepancy and implications for the blah, blah, blah. And without exception, we polled the audience. This was before the days of online polling, by the way. We were pioneers. We polled the audience, and they said that the one without slides was much better. Why? Because that speaker was in the room with the audience. They were engaged. They were connecting. They were using gestures. They were using voice for the first time. Realize that most of you, the moment you lean on the slides, you become robotic in your voice, in your tone, in your speed. You lose contact with the audience. You lose that energy. And I know why. Most of you are afraid of that energy. But realize the energy that you give in a presentation affects the energy of the audience as they're listening to you. So if you have the impression from me, if I were to give you this workshop all the time looking at my notes, looking out the window, whatever, if I did that all the time, you would eventually give up on me as well. And that's just that energy, all right? So you want to be very mindful of that, very aware. And then I think the last part of what I wanted to say is about the question and answer session. This is a lot of stress for a lot of people. And I want to give you 3 key tips. One is breathe, which, by the way, if you have nerves in general, breathing is one of your best medicines. Before you have to stand up to give your talk, breathe. One trick I learned, which can be nice, those of you who do yoga already know about this kind of thing. You already know how powerful just slowing and consciously breathing can change so much of your physiology. You can do this exercise even if you're not a yogic guru. Breathe in and count as you breathe in, and breathe in deep and count. So let's say you really try to expand your lungs and you count to 12, 13. Then breathe out and make that out breath take at least twice as long. So if you were 12 to breathe in, make it 24 count to breathe out. And if you do that 2 or 3 times, sometimes I did this in workshops, and some of the attendees, they would start to, oh my God, I'm going to faint. Because you never do this. You never give yourself that. So just trying that before you get up, for example, trying it. Don't do it during, because if you have your microphone and you breathe like Darth Vader, it might get really weird. And then you'll get even more nervous. So that's a different workshop. But there are medications for that. While you're breathing, when they're starting their question, breathe when they're starting their question, then repeat the question back to the audience. That buys you time and allows you to restructure the question. Because some of these questions aren't questions. They go on, they're actually about the ego of the speaker. So then you say, okay, what I think you're asking is this and this. Yeah, that's basically what I'm asking. Okay. And it also, by repeating the question, you're forced to listen to it till the end, right? Most of you, I know what you do. You get up, you're at the microphone, that famous professor from your field gets up and he's going to ask you a question, and you immediately start to, okay, and then you remember, gotta breathe. Okay, you're hyperventilating, and then they start with their question, and the question takes 3 minutes. But they're only 10 seconds into the question, and you've already formulated 3 defenses. So you stopped listening to them. You heard the first words because oh my God, he's going to ask about that one Western blot. I know that I didn't do the control. I know that, but it's not my fault because my technician screwed up. Now you're on that Western blot, and you've already created 3 defenses about that Western blot. It turns out the guy wasn't asking anything about that Western blot, but you didn't hear him because you were in your own little defense mode. So really listen to the question until the end.
With this, I will take the opportunity to start the question answer session.
Perfect.
We have many of them.
Oh, great.
Thank you for the great talk. One of them, the first one is from Helena. She asks, how do you avoid sounding repetitive?
Look, someone asked me this about a paper. I think that it's harder. It's much harder than you think to be so repetitive that the audience gets annoyed. Okay, fair enough. If you have a 15-minute talk and you're presenting 3 Westerns, and then you're robotically repeating the same thing over and over, then maybe it can get annoying. I think it's hard to be repetitive. The best test for that is just to practice your talk in front of your audience. I'm sure they will give you feedback about that. 9 times out of 10, the problem is the opposite. The problem is people, as they go along, especially that black hole in the middle, they're losing sight of the big picture. If you are worried, here's a general advice for all of you. I would say 2 things. 1, if you're speaking so slowly when you give a talk, that your inner voice, which by the way is almost always negative, that your inner voice is saying, wow, you're talking so slowly. I bet they're going to think that you're retarded or something, that you don't know what you're talking about. You're an idiot. That is probably just about the right speed to speak at. And I mean, my comment applies sort of pre-COVID when there are larger rooms and with a microphone where it's really bad. The second advice I would give you is, which I just blanked on. I have a blank on now. If you're repeating your nucleus enough that your inner voice is saying, maybe this is a little bit repetitive, my guess would be that's about the right level of opportune repetition, so in strategic places. But, really, if it's too much, the audience will tell you. And I can almost guarantee you, even if you really were to annoy them with that, they would still they would kind of learn to fly over it and ignore it, and then zoom in on the things that were new. So the other last thing you can do if you are really worried, just change it up a little bit. So at one point you say, we want to identify regulators, upstream regulators of this protein. Then later on say, our results suggest that X and Y, that these proteins drive the activation of this downstream whatever. Just change up the words a little bit.
Before we proceed to the next questions, is it fine for you to extend the session a bit, maybe 10 minutes, to answer the questions?
Sure.
Okay,
Yes,
perfect.
the children haven't started screaming, so I think we're okay.
Okay, so the next question is from Alexandra Witt. She asks, in a lot of cases, passive constructions make it hard to keep subject and verb together. However, some of my previous supervisors have discouraged use of I or we. Any ideas to work around that?
Right. Great question. So this is a generational change, and I used to have a slide where I looked at so I had a quotation from Science, and I had a quotation from British Medical Journal, and maybe from Nature. I forget which one of and journals from other fields. So when I was young, okay, much younger, you really were expected to write passive voice, and using I and we in natural sciences was, I would say, quite unusual. There has been a sea change. There's been a generational shift. So if you go to these journals, these ones I mentioned, for example, I found quotations from their editorial boards, and they tell you, please use active voice. Please use I and we. And they'll tell you why. They say, because this leads you to write shorter sentences that move better. So anyone who's now 50 or older, 60, this is very different for them. They're very probably uncomfortable. And also there's even a cultural. So in Anglo-Saxon, so UK, Canada, US, for a long time, I think scientists there have been comfortable using I and we, whereas in old Europe and maybe more traditional environments, they're more restrained. Okay, see if you can find an active construction without I or we. So you could say, these results suggest, our Western blots indicate that. Now, some people, you'll find traditional reviewers who say, a study, for example, cannot suggest, or a blot cannot suggest because it's not a person. A person suggests. These are such minor. Do not worry about this. If the reviewer makes a comment, then you know who you're dealing with and you can change it and go to passive. It doesn't matter. But try to make active constructions subject, verb, object. That said, in the methods especially, I mean, I use passive plenty. Just try to keep things simple. Try to keep the sentences simple. Try to keep the thing and the verb kind of, the mixture was incubated. Try to keep that was incubated relatively close. Yeah.
The next question is from Benam Karami. I hope I pronounced it correct. He asks how to visualize very abstract concepts in that how to visualize in very abstract concepts. In that case, we might be inaccurate in some definitions and lose or distort some basic ideas. What is the trade-off between simple visualization and sticking with abstract basic definitions?
That's a great question. I would say the questioner, you're worried, to the questioner, you're worrying a little too much. I'm not proposing, I'm not recommending that you, for example, try to reduce things to metaphors and you say, the feedback loop in this signaling pathway is like when a car runs over a hamster, and then you're really stretching things and you're simplifying and you're distorting. I don't mean that. What I mean is actually to zoom in to each sentence and looking for vague expressions that don't immediately create an image. So for example, if you say, this protein may help regulate, okay, protein A may influence protein B independently of protein C. It's already kind of clear, but realize you could also say, so protein A, we could also write we could change influence to something more concrete, maybe, if we can. You could say influence maybe to activate or maybe inhibit, depending on your result. So you could say protein A activates protein B even after we knock out protein C. So I'm in one sentence, I'm giving you what I said earlier in one sentence, but now I'm even giving you the actual experiment. You're able to visualize how I got to that result. So that one sentence shows not only the conclusion, kind of, that protein A influenced protein B independently of protein C, and it even narrates the key experiment that does it. That's what I'm talking about being concrete. I literally want you to write about your findings so that I can imagine what's being pipetted into the different wells. Because if that's the level where you're talking, yeah? So that's what I mean. That's what I'm talking about. So you don't want any distortion. On the contrary, you want to get maybe technical. But just realize when you get very technical and detailed, then you're going to need to zoom out periodically. I didn't mention this, but listen, everyone. A good scientific storyteller is constantly zooming in and out like a camera lens. Very detailed, bigger picture. Very specific, bigger picture. You have to do that constantly. And a good communicator does it in a way that's smooth so that you're almost not aware that it's happening. So that even within one sentence, you go from metastasis to a particular protein, to an amino acid on that protein, and then to something, maybe colonization of cells. Maybe even in one sentence, or certainly in one paragraph. You're able to do that without the reader saying, wait a minute, arginine 221, what protein are we at? What process are we in? That's a good speaker. They're zooming in and out and keeping you on that thread.
There's another quick question from Kishore Kumar. He asks, is it a good idea to submit manuscripts to bioRxiv database before submitting a peer-reviewed journal?
Talk about getting specific, general to specific. That's a very good question. And if you want, so it's an important question because this is, it's becoming more and more common for life scientists to deposit in preprint archives. This was already done in certain parts of life science, like C. elegans, and this was already done for a long time in, say, physics. But now in even competitive life science fields like cancer biology and so on, and neuroscience, it's getting done. The answer is you have to check with journals. So my best advice, and this was an issue where I used to work at an institute, is there's a Wikipedia page, and again, write me and I can try to send you the link, where they give you a list by publisher of what the a policy of that publisher is regarding bioRxiv and other preprint archives. Most publishers generally have no problem with bioRxiv. One or two might, but most shouldn't. But basically, if you're thinking to put it in bioRxiv, look at where you might hope to submit the paper for the first two times or so. Make sure those publishers would be okay with that.
With this, the last question, which is from Manjula and also me. Do you recommend any good resources to learn more on academic writing for publications you taught us today? Maybe it would be either you could talk about it now, or it would be great if you could put them in your slides and we could refer to them.
Yeah, you're good. Yeah, so right, on my slides, you still have, oops, hang on. So this is this graphic, kind of, it's on a slide, and then at the top is this URL here. So my firm has a website with a page on resources where we have put together several things. That's the first place I would send you. There are websites. I mean, if you just Google Nature, you ran a series of stuff online. I would Google science writing, and you'll find a lot of things. And you have those MOOCs, those massive online courses that you can take. There's really a lot out there for biosciences. That would be my best. And there's also, we have a YouTube channel. So if you go to our website, there's also other YouTube videos. And other people have YouTube videos, yeah. And then contact us, and we can do a training or a seminar or do one-on-one with you.
Okay, then with this, I think it's time to shut the session. Thank you very much for the great workshop. It was.
Well, thanks to all of you.
I think it was very interactive, even though it was all online. It was great. And
Great.
we've got a lot of great feedback. We will get back to you with, for the participants, we'll get back to you with the slides. And I
Yes.
hope we all can incorporate the suggestions we got today.
Good. I hope so. And keep in touch.
Yes.
Take care. Thanks very much to all of you.
Thank you. See you all tomorrow.